Thank you. Today I'm going to present data from a project that was proposed by the Desmoid Tumor Research Foundation. So it's well known that there are beta-catenin stabilizing mutations in the vast majority of desmoid tumors, and these result in beta-catenin that's localized to the nucleus and is thought to drive the neoplastic properties of this tumor type. Beta-catenin plays a role in the canonical Wnt signaling pathway. Normally, beta-catenin is phosphorylated by sites encoded by exon 3, and it stays at a very, very low level inside the cell. When an appropriate Wnt ligand is present, it stabilizes beta-catenin by inhibiting its phosphorylation. The stabilized beta-catenin binds to TCF, LEF transcription factors, gets into the nucleus, and transactivates transcription. This is an architectural transcription factor, so it changes the expression of multiple genes in the cell. In desmoid tumors, there are mutations that delete the phosphorylation sites encoded by exon 3 in beta-catenin. And the two most common mutations that have been identified are the T41A and the S45F mutations. And so the Desmoid Tumor Research Foundation put out a request for applications to generate an animal model in which these two mutations, T41A and S45F, could be activated as a model of desmoid tumors. Ozgene, which is a company that generates genetically modified mice, was a recipient of the proposal to be able to generate these mice, and they went ahead and developed two mouse models. So this is a diagram of the beta-catenin gene. At the top is the wild-type gene. There are 15 exons, and you can see in exon 3, they put a little mark where the phosphorylation sites are. The way they generated these mice was to put the mutation into the normal exon. So they took exon 3 and they added in either the S45F or the T41A mutation into that site in exon 3. And then before exon 3, between exons 2 and 3, they put in a wild type, a cDNA for beta-catenin running from exons 3 to 15, and they surrounded this by LOXP sites. And so the way this works is when the cells are exposed to Cre recombinase, the two LOXP sites recombine, the wild type cDNA is gone, and instead the cells produce the mutant form of beta-catenin, which is exactly genetically identical to what we would see in a patient who has these two mutations. So they generated these mice, and we've been looking at determining what the phenotype is and determine if they function the way they should. So the first set of experiments that we did was to take cell cultures from the mice and then activate the conditional alleles using an adenovirus either expressing Cre recombinase or control GFP. And then we looked at the cells by Western analysis to see whether or not the right proteins were being made, and by RNA to see whether or not beta-catenin signaling was activated. And we found that for both of the two mice, when Cre recombinase was put into the cell cultures from the cell cultures of the mutant mice, we saw a lack of expression of the S45 or the T41 phosphorylation site in those mice. And this shows that when you stabilize mutation, you knock out those sites, and that epitope, those phosphorylation sites, are gone. We then went and looked at the expression of axin 2, which is a beta-catenin target gene in fibroblastic-like cells, and found that when these two genes were activated, the two mutant genes were activated inside the cells, there was an increased expression of axin 2, showing that we were able to activate beta-catenin signaling inside the cells. We then went back to the mouse and wanted to determine if these mice would develop desmoid tumors. So to do this as a start, we injected an adenovirus expressing Cre into the leg muscles into the mice. And this is an approach that had been used by Tyler Jacks to activate mutant forms of P53 and KRAS inside mice to develop sarcomas. So we took the mice and we injected an adenovirus Cree into the leg, and then we went to observe them to see whether or not they might generate desmoid tumors. However, this is not always the best way to be able to cause recombination, and you're not always sure what kind of cells might get the recombination, might get the Cree recombinase adenovirus into them. So the second approach, we use a genetically modified mouse in which we could turn on the Cree recombinase by injecting in tamoxifen, and we use this mouse, injected tamoxifen into the limb, and then want to see whether or not these mice developed desmoid tumors. So far for the ones that we injected adenovirus Cree into the mice, so far we have not identified tumors, but we also did not identify cells that had recombination. And so it's quite possible that this approach to being able to turn on the mutant cells in vivo is not very effective. We have done the work as well on the tamoxifen-based system. Unfortunately, we started this right before the COVID hit the United States, and during that time we had to close our labs. We had to sacrifice a number of our mice, and so we don't have any data from this yet. And so we restarted this as the COVID slowdown passed, and we've regenerated the mice, we're recrossing the mice, and hopefully we'll have some data from this in the next couple of months. So I can tell you that Gosdine did generate these mice. The in vitro and cell cultures, adding Cree to the cells will cause recombination and will show expression of the mutant forms of the beta-catenin and will activate beta-catenin mediated signaling in the mice. We don't yet know if these will develop desmoid tumors, but the mice do work for activating beta-catenin. Our intent is to make these mice publicly available, and once we can show a phenotype, we'll deposit them in Jackson Labs. In the meantime, however, if any investigator anywhere in the world wants these mice, please write to me, and I'd be happy to share them. So these can be available, and we'll send the information on how to do genotyping, as well as more of the mapping information about the specific mutations and where LOXP sites are, if anyone's interested in using them or obtaining the mice. So hopefully next year at this time we'll have mice that actually develop desmoid tumors. I'm disappointed that we don't have these yet, but we're really slowed down quite a bit by COVID. But we do have mice that will express these mutations they are able to be activated by Cree recombinase. So thank you for your time, and I'm open for questions.